This is a quick review on Chapter 17, Gene to Protein. And oftentimes in biology, they call it the dogma, that we have DNA, we have a specific gene, and it codes for, the DNA codes for a gene, and it codes for a protein. So that's the central dogma behind biology, that we have that gene to DNA, and we eventually are going to get our protein. So we're going to go over, first of all, scientists aren't in this test, this Chapter 17 test, so we're going to concentrate on how DNA is used to make a protein. And it starts in the nucleus, where we have our double strand of DNA. And again, our leading side, that 3 to 5 side of DNA, is all that is used to make a protein. So we have RNA polymerase is going to come and help get to what we call the TATA box. That is the code in DNA that would be a thymine, an adenine, thymine, adenine. And after that, we're going to have a start code A, and that's where messenger RNA is going to start. They're going to read 3 to 5 end, but they're going to build 5 to 3. So if here I had my first 3 pieces of code, and let's say it's a G, a C, and a G, then messenger RNA is going to say I need the opposite code of that, a G, a C, and a G. And that's what you did on your transcription worksheet, is that you worked with, if you had the DNA code and you had your 3 pieces of code on your 3 letters, then messenger RNA is going to use the opposite code of that. So that's what it does. So messenger RNA starts making a strand, and all of those has a start code on it. It'll have all of the amino acid codons, the 3 little letters that we need, each creating one code for one amino acid. And then in between that, though, are pieces that we don't need. Those are going to have to be removed. So the first part is they'll find the promoter area, and your RNA polymerase is going to come in there, and it's going to start synthesizing down. It'll start building the code from the 5 to the 3 end. And once it hits a stop code, it stops that. So there's one start code, AUG, and there's 3 different stop codes. Once it's finished, the messenger RNA knows it has all of the code it needs to make a specific protein. So now it has to, transcribing it is done, and now we're going to have to go through the processing piece. And in the processing piece, that is where the introns, or the pieces that we don't need, are going to be spliced out by spliceosomes and removed, leaving only those, the exons, or the expressed pieces. But then it's too dangerous to bring that through the nuclear membrane, so they have to protect it. So they have to put on a poly A tail and a guanine cap on that. So here you can see the little cap and the tail. And once that's done, the code has been read. Messenger RNA has made the opposite code. All of them are codons, those little 3 groups of letters. Remembering that any place in the DNA that they found a thymine, they replace that with a uracil. RNA cannot deal with any thymine, so it's all uracils. And then it's going to leave the nuclear, through the nuclear membrane, and it's going to go down the endoplasmic reticulum to the site of a ribosome. And that's where translation happens. So the first 3 pieces of code, your start codon, comes through, and then the next 3 letters, we have a code, but tRNA is going to go and get the opposite code of that. So that's called the anticodon. And again, referring back to your transcription worksheet, what we end up with your anticodon is the identical code we had from DNA, except that any time that there was a T in the DNA, they substitute it for U, so for uracil. So as the 3 pieces of lettering come in from messenger RNA in here, then tRNA gets the anticode, or the opposing code, which match the DNA strand. And eventually they'll end up with a stop code, and that stop code indicates that all of the amino acids that are needed to make a specific polypeptide chain that makes a protein is done. And so then that would end up leaving and going to wherever else it's needed. So again, we have our start code. We have the TATA box, the promoter region, and the start code. And then the nucleotides, mRNA, is going to bring in an opposite piece of nucleotide. and every 3 letters from that DNA is called the codon. So your pre-mRNA is going to have a start code, a series of codons, and then a stop code. In the RNA processing, they spliceosomes remove any piece that isn't necessary to make an amino acid. Those are called your introns, leaving only your exons. But before it can leave, to make sure that that messenger RNA strand is safe, they put on one end a poly A tail, and at the one end a guanine cap. And that keeps the 2 ends safe while it's transitioning through the nuclear membrane and to the ribosome. And then methanone, your AUG, is always your start code, and so that would come into the side of the ribosome, and then tRNA is going to start bringing in the anticodon, or the opposite 3 letters from what you have, and you end up with that polypeptide chain. And and of course in here we have, with your transcription, we have the initiation where RNA polymerase binds to the promoter region. It doesn't have a primer here, it has a promoter region. And then the messenger RNA elongates as it's making that big long strand. And finally, when we get to the stop code, that is your termination. So in transcription, we have your initiation, where the Tata box in your start code is going to be. We have elongation, where the nucleotides, RNA polymerase is bringing in nucleotides opposite to the DNA code. And then termination is when we get to the stop code. Once the introns, then we have RNA processing. Once the introns are removed and the tail and the cap are put on, they go to the ribosome for the translation. And during this piece, we have 4 major issues that we can have. And again, remember that the RNA polymerases are supposed to be making sure that there are no errors, but there are sometimes errors, and there are 4 of them, and it's important for your test that you know the 4 errors. The first is called a frame shift mutation, and that is when there's either a deletion or an addition of a DNA nucleotide that does not add or remove 3 nucleotide bases. So in other words, messenger RNA is produced on that DNA template, and it's read in the codons. But if we have an extra, if mRNA reads by mistake the fat cat ate her hat, instead it reads it incorrectly, and you get the ATC, ATA, the ERA, AT, instead of cat ate her hat, then you're going to have an error, mutation, and usually it will be a non-functional protein, and it would be destroyed. The second one is called a missense mutation. And this is really important because missense is what is, as I told you in our Google Meet, is responsible for sickle cell anemia. So what happens is that a wrong nucleotide is placed in. It's not that they're shifted over like in makeshift where 1 letter is mistakenly shifted, and that's all it takes is just 1 letter in makeshift. In the missense, a wrong nucleotide is put in. And so for just 1 mistaken nucleotide, it can cause the protein in blood to be abnormal and be sickle-shaped instead of a nice round circular piece. And so, and of course, you know, that's one nucleotide substitution, so that's a missense. And again, remember, missense is what causes sickle cell anemia. A nonsense is when a wrong nucleotide is put in the sequence. And usually that ends up with a premature stoppage. The RNA polymerase usually recognizes that it's incorrect. It's oftentimes there'll be an early stop code put in, and it tells the protein synthesis to stop. So, and again, that just like in a frame shift mutation, that would be in a protein that would not be functional. So far we have 3. We have frame shift, that the protein would be non-functional. Unfortunately, missense, the protein is functional and causes a disorder, sickle cell in one case. There are many others. Nonsense, again, it would be a non-functional protein. And then the last one is the one that I tell you is my issue, where it's called the thymine dimmer. And that is with age and heavy amounts of UV light, or your very pale skin and your a body doesn't deal with the UV light very well, you have mutations happen with the epithelial or skin cells. And the thymines will then glue to the next nucleotide base, and it can't be read correctly. And what happens is that is a skin mutation that will cause several different types of skin cancers, basal cell, melanoma, etc. So the thymine dimmers are what are causing, those are environmental errors, and they cause issues for you. So again, this is a quick review of chapter 17, and I would pay I would spend a lot of time on how transcription happens with your, goes to the initiation with the promoter, your TATA box. We then have elongation of the messenger RNA. And then finally, and that's 5 to 3, and on your leading strand, or your template strand. And then finally, you're going to have one of the 3 stop codes that will terminate it. So that's transcription. You then go through RNA processing, where the introns have to be removed, a poly A tail is placed on, and a guanine cap is put on. It then leaves the transcription, leaves the nucleus, and it goes to the site of the ribosome for translation, where then tRNA goes and gets the 3 opposite pieces of code, the anticodon, and those are what code for the amino acids. And so you'll have an amino acid chain of one amino acid after another, and that's what makes your polypeptide or your protein. And that's it for our review. Thank you.